Hello everyone, this is the demo of Simcyp Biopharm Software, and in this demo I will be taking you through Tacrolimus amorphous solid dispersion formulation. So in this demo we'll be looking at how the software can be used in setting the specifications, performing risk assessment, and also how the software could be used in formulation design. So before going into the demo, a brief background on Tacrolimus. It is a BCS class 2 drug used as an immunosuppressant. Being a BCS class 2, it has low solubility and high permeability. So the crystalline form, which has a solubility of about 1.8 microgram per ml, which is very low. And therefore, what was done is it was converted into amorphous solid dispersion, where the amorphous form is having very high solubility compared to the crystalline form. But the disadvantage of these products, amorphous solid dispersions, is sometimes on storage, they could slowly convert back into the crystalline form. And if this conversion happens, the solubility would be reduced, thereby bioavailability and therapeutic efficacy. On the market, we have several generic formulations for tacrolimus, and one of the generic formulation was susceptible to this crystallization on storage at accelerated conditions. This is a graph of percentage crystallinity versus time at accelerated conditions. So as you can see, with time, the percentage crystallinity of the drug product is increasing. So in this particular demo, we will be seeing, or we will try to predict at what percentage of crystallinity the drug would be by an equivalent to the reference formulation. To do that, now we will be switching to the Biopharm software. So the software itself is quite similar to the main simulator, where we have population specific data, compound specific data, and trial design specific data. In the population, we provide two default populations: the healthy volunteer population, which are based on North European Caucasians, and pediatric populations. In the population screen, you can find basic demographic information. You also will find tissue composition and tissue flow rate information, which are used in predicting the distribution of the drug. And also, we provide additional physiology data for liver, kidney, brain, lung, and tumor, which are used in the permeability limited models of these organs. And finally, we have GA tract specific data, which is a huge physiology database having all the parameters which could influence the solubility, dissolution, metabolism, and absorption of the drug. And in the compound data, we have PhysChem screen. In the PhysChem screen, we have to input the molecular weight, log P, pKa's, uh, blood to plasma partition coefficients, and plasma binding as well. So tacrolimus is a high molecular weight, high log P molecule, and it is a neutral molecule, and you can select the neutral molecule from this drop-down. So once you define the PhysChem properties, what we can do is we can go ahead and define the absorption model. So in the absorption screen, you can find a simple first order absorption model. However, for biopharmaceutical applications, we use advanced dissolution, absorption, and metabolism model called as ADAM model. And if you have special cases, like say, for example, if you want to model drug absorption through lymphatic route, or if you want to model the basolateral transporters, then you can use another model called multi-layer gut wall ADAM model or MADAM model. However, for the case of this demo, we are using ADAM model. And in the ADAM model, the main important parameter predicting the absorption is permeability. So we provide We have correlation-based models which can be used to directly use the in vitro measured uh, cell line permeabilities. However, for this particular case study, we are using a mechanistic model called McPef model. So what this model does is it is based on first principles. So mechanistically, it predicts the permeability in different parts of the GI tract using FISCEM data, or it can be also calibrated using in vitro measured permeabilities. So the main advantage of this model, apart from the fact that it can predict uh, permeabilities for different parts of the GI tract, is that it can take into account the drug binding to the bile muscles or excipients. So what happens is if the drug binds to bile muscles or excipients, the free fraction of the drug decreases. When the free fraction of the drug decreases, the permeability of the drug decreases. So this model can be used where you see negative food effects or the cases where your excipient is decreasing the permeability, then you have to use the McPef model. So in this case for tacrolimus, we are using the McPef model for predicting the permeability from FISCEM. Moving into the formulation screen, here you can define the formulation type. We have solution formulations, suspension formulations, immediate release formulations, entry-coated, and controlled release formulations. Tacrolimus is an immediate release drug product, so we have selected immediate release. And once you select immediate release, what you can do is you can directly input the dissolution profile, or you can mechanistically predict the dissolution within the Biopharm software using a diffusion layer model. So in the diffusion layer model, we have two models which can be used for predicting the dissolution. One is mass balance only model, and the other one is particle population balance model. So this particle population balance model is more advanced model, and what it does is it tracks the number of particles going from each segment of the GI tract to another segment. So the number of drug particles are tracked when we are using the particle population balance model. But when we use mass balance model, we only track the mass of the drug which is going from one segment of the GI tract to another segment. The main advantage of using particle population balance model is it allows us to use two solid state option. So this two solid state option allows us to define what percentage of uh, uh, amorphous form or crystalline form are there in a given drug product. So for example, as we discussed in case of tacrolimus, it has a tendency to convert storage. So the amorphous form could be converting into the crystalline form. So using this model, you can simulate uh, the percentage amorphous form or percentage crystalline form in the dosage form. So you can specify that the dosage form is containing 90% amorphous form and a 10% crystalline form. Or you can specify the dosage form is having, say, 100% amorphous form and 0% crystalline form. So this model, population balance model, would really help us to simulate these kind of cases where we have multiple forms, crystalline and amorphous, or salt or free form in the dosage form. So once we set up these, the next thing what we have to do is we have to input the solubilities of amorphous form and also the crystalline form into the software. So the amorphous form being high soluble than the crystalline form, it has a solubility of 50 microgram per ml. So we input the intrinsic solubility over here in this box. And for the solid state 2, which is the crystalline form, we input the crystalline solubility of tacrolimus, which is about 1.8 microgram per ml. Once we input the solubilities, we have to define how well the drug can partition into the bile muscles to predict bile muscle mediated solubility of the drug. So for this, the software can predict the partition coefficients of the drug into the bile muscles, or if you have data on the partition coefficients, you can directly input the data over here. Once the bile muscle partition coefficients are selected or predicted, then you can define the supersaturation and precipitation of the drug within the GI tract. So for supersaturation and precipitation, we have simple first order models, or we also have advanced precipitation model, which is based on classical nucleation theory. So for this case study, we will be using the first order models to predict the precipitation of the drug in the GI tract. And another important aspect which we have to consider is the conversion of the amorphous form into the crystalline form within. Sometimes when you dose an amorphous form, once it goes into the stomach or small intestine, what could happen is the amorphous form could convert back into the crystalline form inside the GI tract. So when this happens, you can select this option called precipitation to solid state 2. So where you can say that the solid state 1 within the GI tract could precipitate to solid state 2, which is the crystalline form. So for the case of tacrolimus, we are not doing that. So we are not saying that the drug doesn't convert into the crystalline form in the GI tract in case of tacrolimus. And it was due to the fact that in vitro studies, it was observed that the tacrolimus amorphous form is pretty stable in the in vivo conditions. And this is due to the excipient HPMC, which is a precipitation blocker. And once the absorption model is set up, for tacrolimus, we have selected minimal PBPK model for predicting the distribution. And for elimination, we have selected enzyme kinetics, inputting the Vmax Km of the enzymes responsible for tacrolimus metabolism. Once the model is set up, what we can do is we can use the virtual bioequivalence module to predict the effect of crystallinity on the bioequivalence of the drug product. So the virtual bioequivalence module can be activated through this option on the top ribbon. So once you activate, you would have a screen like this, where you can specify the population in which you want to run the bioequivalence. And you have to load two compound files. One is the reference compound file, and the other one is the test compound file. So in the case of tacrolimus, we load the same tacrolimus compound files as a reference formulation and also test formulation. But the only difference we make is the percentage crystallinity in these two formulations. In the reference formulation, it would be a 0% crystalline formulation, that being amorphous form is 100%. And in the test formulation, for this case, we are inputting 20% crystallinity and 80% amorphous. So once you specify this, you can go to the trial design option to specify the bioequivalence trial. So over here, you can select either a general crossover trial or partial replicate trial, full replicate trial, or parallel trial. So for this case, we have selected a cross 2 into 2 into 2 crossover study. And then we can specify the number of subjects which are required in the bioequivalence study. So once this is done, the another important aspect of bioequivalence, virtual bioequivalence specifically, is the within subject variability. Because we are performing a crossover trial, so in a crossover trial, what we do is we take the same subject and dose the test formulation, and after 2 weeks or 3 weeks period, we dose the reference formulation. So in single subject, we are dosing two formulations to perform the bioequivalence. So what could happen during this period is, say for example, the some of the GI tract parameters like pH, bile salts, they could change from one period to another period. And this change in the subject parameters is called within subject variability. So defining this within subject variability is very important because it defines how the confidence intervals are in your virtual bioequivalence study. If you have higher within subject variability, you would have very wide confidence intervals. And if you have less within subject variability, you have very narrow confidence intervals. So in this Biopharm software, we take into account that these GI tract parameters change from one period to another period when performing a virtual bioequivalence study. Once we specify these, we can go ahead and specify dosing regimen. So for the Tacrolimus case, we have selected fasted study, but you can also select fed study and you can also select food staggering model. So food staggering model is a great model which can what it can do is it can reproduce an actual bioequivalence study. So for example, in an actual bioequivalence study, you are dosing in a fasted state with no meal. Then after 4 hours, you are giving food. You can specify these kind of conditions using the food staggering model, reproducing the real bioequivalence cases where in a fasted study after 4 hours, you have to give food to the subjects or healthy volunteers. So this model allows you to specify these kind of events when you perform a bioequivalence study. And sometimes this is very useful because when you give food at, say, for example, 4 hour interval, due to increased bile salt secretion or increased blood flow rates due to the food intake, you sometimes see double peaks in your plasma concentrations, and using this model can recover those double peaks in your virtual bioequivalence trials. And then you have to specify the assay of your test product and reference products, and also UOD of your test product and reference products. So once this is done, we can run a virtual bioequivalence trial. And for this case, I have already run the virtual bioequivalence study, and this is the output for 80% amorphous formulation versus 100% amorphous formulation. So this orange curve is the plasma concentrations of the test formulation with 80% amorphous form and 20% crystalline form. And this blue curve is the plasma concentrations of the reference formulation with 100% amorphous form and 0% crystalline form. And as you can see here, when the crystallinity of the drug product is increased to 20%, there would be a failure of bioequivalence. As you can see, the CI limits are less than the acceptable range, both for AUC and Cmax, even though it is passing in case of AUC infinity. Uh, so these kind of studies are really useful in setting specifications. I was a formulation scientist myself in my previous organization and worked on amorphous solid dispersions, developing the formulations. And thinking about the modeling which is available now, the modeling capabilities which are available now, it would have saved us a lot of time and effort in setting specifications and performing risk assessment and in general understanding the molecule and understanding the formulation as well. Uh, so the case study is done here, but I want to quickly show you a few of the other uh, aspects of the Biopharm software, which I think are very interesting. So one of the important aspect of this software is if you have a drug which is either acidic, basic, or ampholyte. So sometimes the formulation scientists or formulators, what they do is they generate a salt form of the drug to increase the solubility and dissolution. Uh, so we have developed models to predict the solubility and dissolution of salt form of the drug. So we use the KSP-based models to predict the solubility and dissolution of salt forms, and we also have models to mechanistically predict the surface pH of the salts, which drive the solubility and dissolution of salts. So if you have a formulation with salts, this model would be really useful in predicting the solubility, dissolution, and absorption of salts. And we also have model effect of excipients. Say, for example, if you have added a binding excipient like cyclodextrin or polymer binding excipients, what you can do is you can use the excipient model of the software, and you can predict the effect of these excipients on the solubility, permeability of your drug. So we have provided the models for surfactants like SLS, twins, and generic surfactants, and also for binding excipients. So for a formulation design, these models could be really useful in understanding the product and designing the product. And finally, in addition to the models which I have shown you, we also provide mechanistic IVIVC option within the Biopharm software, which can be selected on the top ribbon. And compared to the conventional IVIVC, the main advantage of mechanistic IVIVC is that it takes into account the gastric emptying, permeability, uh, the first pass gut wall metabolism, transporters, and first pass hepatic metabolism during deconvolution step, and it gives you pure in vivo dissolution, which would be very useful in developing good IVIVC correlations between in vitro dissolution and in vivo dissolutions. And finally, with when you have a good IVIVC correlation, it could be really useful in setting specifications or in formulation design. So to wrap up, what we have looked in this demo is we have looked at a case study of tacrolimus amorphous solid dispersion, and we have looked how the software can be used in uh, performing risk assessment and how the software could be used in setting specifications for the percentage crystallinity in the tacrolimus amorphous solid dispersion formulation. And also, we briefly covered upon some of the interesting models which could help a formulation scientist or which could help that product design using the biopharmaceutical software. Thank you for listening.